Mic check.
Mic check.
Hello, guys. Welcome to the Antibuddies Journal Club. This is our 17th episode, and joining us today is Dr. Martin Stacey.
Hi.
Let me quickly introduce Martin. Martin has had a long term interest in molecular and cellular mechanisms of the innate immune system. And since his appointment as a lecturer in immunology at the University of Leeds, UK, his lab has been focusing on epithelial cell biology and inflammatory skin diseases. And he also told us that he enjoys running, cycling, or anything that allows him to escape his two children after after a year of COVID-19 induced homeschooling. I don't have children, but I empathize because I've got a dog and she's always asking for attention whole day.
Yeah, I think children may be even more irritating than
Even
dogs.
more irritating, yeah. That's like all right. So Martin, I'm going to hand over the stage to you. You should have the screen sharing rights.
Okay. Um, Okay, well, thanks for that kind introduction, and thank you for the opportunity to discuss some of the data that we've recently generated in our lab and that we've managed to publish in the December edition of Cell Reports last year. Now, the vast majority of this data was actually generated by two postdocs in my lab, Tom McLeod and Joe Ainscroft, and I have a long-term collaboration with Dr. Miriam Whitman, who I did a lot of this work with. Okay, so my lab has a long-term interest in immunology, and more recently, we're very interested in the immune response of epithelial tissues. Now, if you consider where epithelial tissues are and what they do, then you'll soon realize that they form a critical interface between us, the host, and our environment. And they're involved in multiple different physiological settings. So if you think we have our lungs, which are absolutely crucial for gases exchange, we have our epithelial layers in our gut, which are absolutely crucial for nutrient absorption. And then we have our sensory organs, such as our eyes, our nose, our mouth, etc. And effectively, in order for them to carry out their vital functions, they have to, or very often have, semi-permeable structures, and they're in constant contact with our environment. Okay, so one of the problems with that is because they're constantly involved and constantly interacting with our environment, we can potentially come across various different pathogens. So we can come across pathogens such as Streptococcus pyogenes that give us throat infections. We can have skin infections such as Staphylococcus aureus or Trichophyton rubrum, which causes athlete's feet. Or we can have Aspergillus infection. So most healthy people will not actually have Aspergillus infections, but we're constantly inhaling the spores from Aspergillus. But if we have some sort of chronic disease or immunodeficiency, we might get aspergillosis. We may have helminth infections or E. coli sitting in our gut. And we may also have fungal infections of our urogenital tracts. Okay, so we have all of these potential pathogens. However, in terms of numbers, we're constantly surrounded by, and we have lots of commensal bacteria living in and on us, and these greatly outnumber the actual amounts of pathogens that we actually come across. So in reality, we need to have some sort of way to discriminate the difference between harmful pathogens, so these guys over here, and the harmless ubiquitous microbes, which are absolutely everywhere. And once our body has actually recognized or discriminated between them, they need to be able to elicit an appropriate response. Okay, so the whole process needs to be highly regulated. So if we have an insufficient response, then we may get diseases such as necrotizing fasciitis. So this is called by strep pyogenes. So if we don't have a good response against that, we can have nasty flesh-eating diseases. Also, if we have genetic mutations or SNPs or single nucleotide polymorphisms within our genome against certain entities, let's say of fungi, then we may also get chronic diseases as well. So one of the examples of this is the Dectin-1 gene. Now, Dectin-1 is responsible for recognizing and binding to beta-glucans, which is a cell wall component of fungi. And individuals that have defects in this particular gene may suffer from chronic mucocutaneous candidiasis, which, as you can see, doesn't look very pleasant. So this is what happens if you have an insufficient response against a microbial insult. But on the other flip side of the coin, you can have an excessive response or too much of a response against things, and this can cause inflammatory damage. So one good example of this is the syndrome called DIRA, or Deficiency in R1 Receptor Antagonist. And this is where you have a defect in a negative regulator of the inflammatory response, and you get aseptic inflammation, so you can get pustules or systemic inflammation in various parts of your body. And this is due to the lack of a negative regulator of inflammation. Also, there's a variety of skin diseases, so one of them being plaque psoriasis. And this disease is actually caused by an overzealous immune response. So you get a Th1, a Th17 response, which causes an inflammatory response, but also it has a large innate component and interplay with keratinocytes from our skin. So again, this is an example of where you have too much of a response. Okay, so to summarize, we really need a balanced and appropriate response, and this is absolutely critical to prevent both infectious disease or overwhelming infectious insult, but also in preventing an overt, overzealous inflammatory response. And that's really what the rationale behind the paper that we're going to be discussing today is all about. Okay, so the rationale of the paper is really to investigate what the mechanisms are, or what one of the mechanisms could potentially be, that enable these epithelial tissues to determine the difference between something that's harmful, a harmful pathogen, and a harmless commensal. Okay, so there are potentially multiple different mechanisms. I wouldn't like to say that this is the only mechanism, but this paper will focus on the pro-inflammatory cytokine, which is mainly produced in epithelial tissues, called IL-36, which our lab has been working on for some time. Okay, so let me briefly introduce IL-36 and the IL-36 cytokines. It's a member of the IL-1 superfamily of pro-inflammatory cytokines. There are 4 different genes, so these are bona fide different genes. They are the agonists, so IL-36 alpha, beta, and gamma, which cause a pro-inflammatory response. And then we have the antagonist, the IL-36 receptor antagonist, which will actually prevent this pro-inflammatory response by binding onto the receptor. Now, if you look at the receptor, it's a heterodimer, and one of these particular subunits is the IL-1 receptor accessory protein, and this is common to all of the IL-1 receptors. So this is involved in the signaling of IL-1 alpha, beta, IL-18, IL-33, etc. But unique to the IL-36 receptors are the IL-36 receptor, which dimerizes, and then when it binds on to the agonists, alpha, beta, and gamma, will initiate the IL-1 signaling cascade. It will activate the transcription factor NF-kappa B, and this NF-kappa B will cause the transcription of various different pro-inflammatory cytokines. So many of these might be mediators of inflammation, so chemoattractants that will attract neutrophils. So these are IL-8, CXCL1, CXCL3. We have chemokines that will activate Th17 cells or increase the recruitment of Th17 cells, and will also be able to increase the inflammatory response via increasing the Th17 response via IL-23 and IL-17. So also the IL-36 receptors are known to actually increase VEGF, which is vascular endothelial growth factor, and this will increase the number of vessels, which is often seen in diseases such as psoriasis, where you get an increase in vessel formation. So we get a big inflammatory influx, and we also get the production of antimicrobial peptides, which actually kill bacteria via punching little holes in their membranes, and these are the S100 proteins and the beta defensins. So as you can see, the binding and the activity of IL-36, and indeed all of the IL-1 superfamily members, they can really initiate a high amount of pro-inflammatory cytokines and inflammatory responses. So it's particularly important that these are highly regulated.
Hey, Martin, can I add something?
Yeah, sure.
Just I have been thinking of these class IL-1 superfamily cytokines as a kind of danger signals, even though they are not danger signals exactly, because all of them signal very similar to the pattern recognition receptors. They might also have the TIR domain that is known to be specific in TLRs and the IL-1 receptor family. So I wonder if these are a cell's own way to tell you that there is some danger, to give that dangerous context to whatever is happening around us.
Okay, well, I suppose the answer is yes and no. So some of them, especially IL-1 alpha, for example, that is normally inside the cell. Right. So as we'll discuss during this paper, sometimes when you have DAMPs, these are damage-associated molecular patterns that are normally confined onto the inside of the cell. These are only released when you get damage. So IL-1 alpha is a classic DAMP, or sometimes we call it an alarmin. Right. And normally that sits inside the cell, but when the cell is ruptured and IL-1 alpha is released upon damage, then this can actually trigger an immune response. So yes, some of these are indeed found in the cytosol and released only upon damage. So they are in fact sensors of damage. So some are expressed in different tissues, however.
So they are just floating around in the cytosol. They're not storage in any endosomes or something.
Most of them are not endosomal, as far as I know. So IL-1 alpha is sometimes found in the nucleus or in the cytosol. And IL-36 is not endosomal, it's just in the cytosol.
Oh.
But there are many different types of DAMPs which are released from cells, such as ATP, uric acid, etc. All of these are a sign when the host is damaged. These are released, and normally they're actually found within the cell. Others may be found in the nucleus. So HMGB1 is a classic DAMP that is normally found in the nucleus, but when the cell is damaged and undergoes necrosis, then this is released into the extracellular environment. But under normal circumstances, when a cell dies in a controlled fashion through apoptosis, then these DAMPs are not released, but they're removed, or they're chopped up or phagocytosed by phagocytes.
For that HMGB1, just a quick digress there. That is supposed to be a DNA bending enzyme. It helps as accessory in transcription and all DNA modifying stuff. And for that, when I read first time that is also a DAMP, it was very surprising to me that we have so many different receptors for things that we are supposed to have within the cell.
Yes, I mean, I think the vast majority of these particular molecules are probably not evolved to be a DAMP. That they have their own sort of individual function. As you say, that's involved in transcription, etc. I think it's also involved in DNA recombination. And I suppose that the cells have then evolved ways of actually recognizing these. So lots of pattern recognition receptors. So one hypothesis is that they actually evolved to recognize damage to start off with, and then some of them indeed also recognize exogenous microbial patterns. So many of the TLRs, which are the classic pattern recognition receptors that recognize bacterial entities, they'll also recognize host entities as well.
Right. The HSP60 is one, right? That TLR4 recognizes.
Yes.
Okay.
Then you also get high hyaluronic acid and ATP and all of these as well.
My last question for IL-36, is this restricted to epithelial cells, or can other cells make it too?
Other cells do make it, although I think T cells can actually generate it, and dendritic cells can also generate it. But I think the highest expressions, certainly of IL-36γ, are in epithelial cells. There is a problem with the general recognition of the protein in that there aren't very good reagents which are specific. We've generated a monoclonal ELISA which recognizes IL-36γ specifically, but lots of the data that you see in published work is normally based on RNA. And again, because as we'll discuss during this paper, then RNA doesn't necessarily mean protein, and the protein, for it to be functional, needs to be in the right place. So it needs to be on the outside of the cell.
Right.
Okay. Shall I continue?
Oh yeah, let's go.
Yep. Okay, so as we say, that IL-36 and R1 family cytokines are highly pro-inflammatory, so they need to be regulated. Now, there are various different ways in which these are regulated. I've mentioned the IL-36 receptor antagonist and the R1 receptor antagonist, but another way is to actually modify the actual protein itself. So many of these proteins are produced as precursor proteins. So IL-1 will be produced as pro-IL-1 beta, in which it's produced in an inactive form, and it has an inhibitory domain on its N-terminus. And it's only after the processing or the snipping off of this particular domain in which it becomes active. So for IL-1, this is done by a multimeric molecular complex called the inflammasome. And activation of the inflammasome, again, is highly regulated in itself, but ultimately it will lead to the activation of an active protease called caspase-1, which will cleave the pro-IL-1 beta and produce an active form. Now, there is an analogous system for IL-36, but this time it's inflammasome independent, and caspase-1 does not cut IL-36. However, previously we've identified some intracellular proteases, such as cathepsin S, which are able to cleave and activate IL-36. So again, you need very specific nibbling off or processing of the N-terminus to form the active form. And in this paper, we really sort of explore the idea that as well as intracellular proteases that activate IL-36, we explore the idea of whether there are exogenous proteases, so proteases from outside the human host, which are able to activate IL-36. Okay, so in summary, you need this precise post-translational cutting, and without that, they will not become active. And for IL-36, it's inflammasome independent. And this paper will explore the idea that it's an exogenous protease. Okay, so before actually showing or proving that IL-36 really is a sensor or a pro-inflammatory cytokine which is induced by microbial challenges, we first of all, the paper set out to actually look at whether expression is actually increased by different microbes. So initially, we looked at various different agonists, which come from various different microbes. So we used LPS, which is a TLR4 agonist from gram-negative bacteria, poly-IC, which is a TLR3 agonist. So you could pretend this is a virus or a double-stranded RNA from a virus. And then we have zymazan, which is an agonist for the Dectin-1 receptor. And what we found is that when you add any of these and if you incubate these with, this is just an epithelial cell line, then you see an increase in IL-36 with all of these. And again, if you used fixed bacteria, so dead Strep pyogenes, and over time you see an increase in the total amount of IL-36 within the cell. And this is a dose-dependent thing. So if you add more fixed bacteria, then you get a more rapid response. We also tried a similar thing with fungus, so this Aspergillus fumigatus, in which we had fixed canidia and we added different amounts. As you can see, the total amount of IL-36 protein steadily increased during this incubation period. We also managed to collaborate and get some nice histology slides. So this is a histology slide of the skin, cross-section of the skin. This is normal skin here. This is your epidermis. This is your dermis down here. And when you get an infection with Trichophyton rubrum, so this is athlete's foot or ringworm, then in pink here, this is a rapid upregulation of IL-36 right in the upper layers of the actual skin. So from this data, it suggests that the microbes really do increase the expression of the IL-36 within the actual cell. Okay, so we can see that the cells are able to actually express and produce IL-36, but for IL-36 to have its physiological function, it's necessary for it to get outside the cell, for it to be secreted where it can bind onto its receptors. So the next few experiments were initiated to actually try and investigate that. So initially we looked by Western blot, and we looked at different types of microbes. So initially we incubated it with Staph epidermidis, and this is a commensal bacteria. And we found that in the supernatant, there was absolutely no secretion of IL-36. But if you looked in the lysate of the cells, then you could certainly see that there was some IL-36 which had been upregulated. However, when we added a pathogenic bacteria, so this is Strep pyogenes, we saw that fairly rapidly IL-36 was released into the supernatant from the actual cells. And we've just quantitated this here. So if you fix the bacteria, then you don't get much of an increase of IL-36 on the outside of the cells. However, if you have a live pathogenic strain of bacteria, then you get release of IL-36 into the supernatant. Okay, so we wondered how this was being released, by what mechanism. We tried various different stimuli of various different release mechanisms, so things that might stimulate pyroptosis or secretion from the cell. But we in fact found that it actually was due to necrosis of the cell. So the way we investigated this is we looked at the expression of IL-36 in the supernatant, but we also found that this correlated perfectly or very well with the actual release of GAPDH. So this is a cytosolic enzyme which is only released when the cell undergoes necrosis. So this is an uncontrolled form of cell death in which the cytoplasm is effectively released into the actual extracellular environment. So this increase in GAPDH, which sort of correlates with the release of IL-36, suggests that you get release of IL-36 when the cell is damaged or undergoes necrosis. So we did similar experiments with Aspergillus fumigatus, so it takes a little bit longer, but again, IL-36 was released when the actual fungus was alive. So when it was fixed, it wasn't released into the extracellular environment. However, when the fungi was alive, then you get release. And again, this correlates quite nicely with the release of GAPDH. So this is just simply due to necrosis rather than another form of active release or cell death.
To be clear, IL-36 gamma doesn't have a signal sequence, right? So it cannot be secreted.
Okay, so yeah, unlike most secreted proteins, IL-36 doesn't have a consensus leader peptide.
Okay.
So just like IL-1, IL-1 doesn't have a leader peptide as well. So you'd expect normal proteins to go into the ER via the Golgi and then be sort of secreted via the plasma membrane. However, IL-36 and IL-1 alpha and beta, they simply don't have these peptides. So okay. IL-1 beta is released by a process called pyroptosis, in which little pores are formed in the membrane by
it's gasdermin D.
Sorry, gasdermin D. Just slipped my mind for a second. Gasdermin D. Whereas this is not the case with IL-36, because sort of very rapidly you see under this circumstances anyway that the cells undergo necrosis. Okay. So we're not completely ruling out other types of secretion. It's just that they'd possibly be irrelevant because the cells are undergoing necrosis and releasing all of their contents anyway.
Okay, so this could be an alarmin.
Yeah, that would be our argument, is that this is a type of alarmin.
And this trap epi caused the increased expression of IL-36 gamma just inside the cell, not in the supernatant, but within the cell, like the other pathogenic, like.
Yeah, so under normal circumstances, if you get any microbial stimuli, whether they're from a dangerous pathogen or a harmless microbe, you get an increase, but it's only in the cell. So, and it's only released from the cell when you get necrosis.
Okay, so it seems like there's a PAMP reception that is increasing the expression inside the cell.
Yes, that's right. So, yeah, there are various sort of MAP kinase pathways which are known to increase the IL-36 expression. So these at some point must come from a pattern recognition receptor of some sort.
Okay.
Okay, so as we mentioned earlier, it's important, as well as production, these particular proteins need to become cleaved to become active. So although IL-36 is released from the cells by pathogens, is it activated or is it in its active form? So to do this, we did some in vitro experiments in which we produced recombinant IL-36 in a bacterial system. So we purified lots of IL-36 from bacteria. We then added the culture filtrate from various different microbes. We incubated them together, and then we looked by SDS page and thought, was there an active difference in size? So had there been cleavage? And just to check that cleavage was actually productive and producing a bioactive form, we also used a cellular-based system in which we got our cleaved product, we added it to a Hackett cell line, which are an epithelial cell line or a skin cell line, a keratinocyte cell line, and we used the pro-inflammatory cytokine IL-8 as a readout. And what we found is by looking at Strep pyogenes, by Aspergillus, by Trichophytum rubrum, and by Staph aureus, and using this recombinant in vitro system, we found that if you incubated for 3 hours with the culture filtrate, then you'd get a reduction in size of IL-36. Okay, so here with all of these filtrates, you get a reduction in size of IL-36. Now, to make the size obvious on a gel, we actually added a larger domain, a larger recombinant fusion protein onto the N-terminus. So the actual real IL-36 only has 18 amino acids, which is cleaved off it, but we added a sumo domain on here, so you can see what happens if it is indeed cleaved. So not necessarily surprisingly that if you incubate a protein with the filtrate from a load of bacteria and fungi, then you get some sort of proteolytic degradation. So we wanted to see whether this degradation formed the active form. So we took this particular product, this cleaved product, we added it onto our Hackett cells, and again, we looked at IL-8 secretion. So in all of these columns here, you can see that when you add the pro-IL-36 gamma along with the filtrate, then you get an increase in IL-8 production. And we can actually prove that this is specific, because if we add the IL-36 receptor antagonist in this column here, then it completely ablates the actual signal. Okay, so this proves that this particular mechanism is IL-36 receptor dependent. And it's not simply that you're adding a load of bacterial or fungal mush onto the cells and getting a secretion of IL-8. So this showed that there was something in the supernatant from these particular pathogens that was cleaving and activating the IL-36. Now, I don't show it on the slide, but we confirmed all of these particular proteins here. We confirmed them by mass spec, and we found that they were indeed the exact size that you should expect from the active form. So this is at the serine 18 form of IL-36. So it looks like these pathogenic proteases activate IL-36. Okay, so the next obvious question is that within this filtrate, we have some sort of component that is activating IL-36, but what is it? Well, the way we did this, again, we took our recombinant system, we added the culture filtrate, but this time we added various different inhibitors, or broad range inhibitors of proteases, to try and narrow down what the protease could potentially be. Okay, so this is one of the experiments. We added different types of protease inhibitors. We have EDTA, which is a metalloprotease inhibitor, so that'll inhibit that class of proteases. We have E64, which is a cysteine protease inhibitor, and PMSF, which is a serine protease inhibitor. And what you can find that if you add the E64, then it largely ablates or prevents the actual cleavage when we actually add the culture filtrate to our system. So this suggests that there's some sort of cysteine protease which was found to be produced by strep pyogenes that was actually able to activate and cleave IL-36. Okay, and this is just an activity assay that, again, when we add our protease inhibitor, E64, our cysteine protease inhibitor, then it completely or very largely ablates the IL-8 secretion in this particular system. Okay, so at this point, we did some literature searching and some guesswork, and we had a guess that one of the main proteases from strep pyogenes is this SPEB protein. So this is a cysteine protease, which is known to be a virulence factor, which is secreted by these strep pyogenes bacteria.
Was that your first candidate? Like first guess?
In this case, we were quite lucky because that is one of the main secreted virulence factors, which is a serine protease. So this time round, yeah, it was a lucky guess. Okay, so we went on to explore whether SPEB-B activates IL-36. We brought in or generated some SPEB-B protein and tried it as a recombinant form, and that indeed did activate and cleave IL-36. And also through our collaborations with a Japanese lab. Oh, hang on a second. Sorry, this is we did a similar approach on Aspergillus fumigatus. And again, this time, instead of finding that it was a cysteine protease, we found out it was actually a serine protease from Aspergillus fumigatus. And again, we did an activity assay. And again, by adding PMSF, so this serine protease inhibitor, we could nearly completely ablate our secretion of IL-8. So it was a serine protease. And again, our guess was it could be one of these virulence factors, which is secreted by Aspergillus. And one of the candidates was called ALP1. Okay, so one way to narrow it down, we produced the recombinant form of these two proteases, and also through our collaborations with a collaborator in Japan, we managed to actually produce a knockout bacteria, or a bacteria that is deficient in SPEB. So this is our wild type strep pyogenes incubated with IL-36. And as you can see, most of it is cleaved and activated, whereas with the protease deficient microbe, then you don't see any cleavage product at all. And in fact, you don't see any activation of IL-36. So we concluded that SPEB will activate IL-36. We also looked at various different proteases from Aspergillus, and we're able to get our hands on two of the main knockouts, or of knockouts of the main secreted proteases. They're ASP-F5 and ASP-F13. And we found that the knockout of the deficient one in ASP-F5 still cleaved IL-36 just fine, but we found that the knockout for ASP-F13 completely inhibited the actual cleavage of IL-36. So our conclusion was that ASP-F13, or ALP1 for its other name, also activates IL-36. So this one here is a cysteine protease, and this one here is a serine protease. So we didn't have time or the inclination to look at the proteases for these two for this fungus and this bacteria, but we did indeed show that they do have some sort of proteolytic activity. So this was all in an in vitro system in which we have recombinant bacterially produced IL-36, and we add the supernatant from bacteria. To show a slightly more physiological setting, we decided to have, or to investigate whether pathogens could induce the release and the activation of IL-36 that was produced from within the cell. So we generated some cells to express IL-36, and these had a flag tag on them for future manipulation. And then we infected these cells with Aspergillus fumigatus. And then we did an immunoprecipitation assay on the supernatant to actually pull down and actually recover the released IL-36. And what we found in this case is in the lysate, you get IL-36 that hasn't been cleaved up here. And then with wild type Aspergillus, you get a slightly smaller band implying that this indeed has been cleaved. With the ASP-F5, you get deficient, you get cleavage, but in the knockout, just like in our in vitro system of the ASP-F13, then you don't get any cleavage at all. So again, we confirm this by mass spec to see whether the bioactive form was produced. And again, if you've got very good eyesight and good imagination, this is the S18 form of IL-36 gamma, which is the active form. And we also took these pull down IL-36, and we showed that the activity of this was dramatically reduced in the ASP-F13. So this showed that in a cellular system, we could take a whole fungus plus and minus the knockout of a particular gene and show that the release and the activation of IL-36 was reduced in the knockout of the ASP-F13. Okay, so what exactly is activating IL-36? So we've shown two particular proteases, one from a fungus, one from a bacteria that are able to activate IL-36. And we've also shown that these this fungus here and this bacteria here are also able to activate IL-36. So it looks like there's a broad range of pathogens and proteases that are able to activate IL-36. So in this sense, it seems that IL-36 has evolved to have to be very sensitive to proteolytic degradation and activation by a whole range of different proteases.
So I have a question.
Yep.
Do you think there are different sites of cleavage of interleukin 36, or is it like around the same place?
Okay, so for IL-36 to be active, the actual proteolytic cleavage has to be very, very precise. So it has to be cut to generate the S18 form. So if you cut it by one more amino acid, or if you have another amino acid, so you have either the 17 or the 19 form, then that reduces its activity by about 10,000-fold.
Right.
So you need an exact cleavage in an exact spot for it to become active.
So how is it that proteases from different pathogens and different like activity proteases like serine or cysteine proteases do cut in this exact spot? Why do you think it is?
Okay, well, I mean, I'll come on to that later, but if you look at the crystal structure of IL-36, effectively it has a compact IL-1 domain, and then the N-terminus is really a flexible loop region. So it looks like that the IL-36 has deliberately, in inverted commas, evolved a very flexible, exposed N-terminus, which is just really very susceptible to proteolytic cleavage, especially at that site. So you have a very compact IL-1 domain, which sort of starts after, amino acid, say, number 20, whereas the N-terminus is really very exposed and very susceptible to just about, not all proteases, but to a large, broad range of proteases. So in a way, it doesn't really matter whether they are cysteine proteases or whether they're serine proteases, then it's just very susceptible to proteolytic degradation at this particular site.
I understand, thank you.
Okay, so again, just to answer your question, we thought it'd be interesting to look at whether this is an evolutionary conserved process. So we decided to look at the murine system. Again, we thought, well, one, if we can show it in another system, it'll be very important, and two, for mouse models and for live in vivo experiments, it would be absolutely ideal to have a mouse model. So if you look at the actual sequence of IL-36 gamma in humans and in mice, surprisingly, that they're completely different around the cleavage site. Okay, so in the mouse system, for you to have an active IL-36, you need this G13 residue here. And for the human system, you need the S18 system here.
Hey, are those real sequences, gravy smack and this great, or are these placeholders?
Oh no, they are the real
okay.
sequence. Yeah, not just wacky, amusing names.
Gravy smack.
Yeah. So effectively, I mean, as far as you can see, there's no consensus sequence, and they are very different indeed. However, we tested the murine system on our pathogenic supernatants, and the mouse system was in fact even cleaner than the human system. So the mouse system, again, you've got cleavage, very specific cleavage in general, and we mass spec this, and we did a very similar bioassay, and we found that this G13 form, which is the active form, was generated by the cleavage. So again, we've got a completely different sequence here. We've got a bunch of completely different proteases with different specificity. So the answer must be in that the actual overall structure of IL-36 gamma allows this flexible N-terminus to be particularly susceptible around this site here. So this was quite a surprising result for us, but from an evolutionary perspective, it just shows that even if these particular pathogens can downregulate or mutate their protease activity, then it's likely that they can't get around this system. So just about any protease or a large number of proteases will be cleaving this site here. So it makes it hard for pathogens to evade this particular response. Okay, so in summary, we have our epithelial cells. So IL-36 is expressed in the gut, in the skin, in the lung, etc. During normal circumstances or high abundance of commensal microbes or pathogens, then we're going to have an increase in IL-36 production. And this IL-36 is within the cell because it doesn't have a consensus leader peptide or secretion signal. And it's only when you get pathogen-induced necrosis, so when you start to have growth of the yeast into its hyphal form, or maybe the production of various different proteases to actually degrade the cells, then in which case you get the release of the pro-IL-36. This pro-IL-36 is then activated by these secreted proteases to form the mature IL-36. And this mature IL-36 can act in an autocrine fashion on the endothelial cells, and they'll produce various different inflammatory mediators, or it can activate production of other cytokines and increase the infiltration of various different cell types to the site of inflammation. You also get the production of antimicrobial peptides, such as beta-defensin and these S100 proteins, which help protect the epithelial layers. So this can be a good thing in terms of when you get a pathogenic insult, but if you get an overzealous response, so maybe when you get the production of too much IL-36, then in the absence of pathogens or microbes, and then the release of IL-36 as you do in the psoriasis, then you could get a pro-inflammatory response. So in pro-inflammatory diseases, especially with psoriasis and eczema, you do indeed get an increase in the amount of IL-36, and this can also be susceptible to other proteases that are around there. So what are the implications of all of this? Then hopefully I've persuaded you to believe that IL-36 is a global epithelial sensor of pathogenic infection, and the actual location of IL-36 right at the actual surface or the interface of our environment. So if you look in the skin, it's right at the top layer of our epithelial cells. Hopefully it can the location will allow it to act as a sensor of microbes. And because it's sensitive to proteases from multiple different pathogens derived from multiple different domains of life, then it's probably very difficult for these pathogens to evade or evolve mechanisms to get around this. So at this point, you might think, well, why on earth do these pathogens produce proteases if they activate IL-36? But it's been shown that these proteases can be necessary for the acquisition of nutrients or the degradation of epithelial cells to actually get in and become invasive in the first place. So these proteases are fairly essential for the pathogens to actually become pathogenic as well. So as I say, yes, so some of these proteases are virulence factors absolutely required by the pathogens for them to actually sequester nutrients, etc. But interestingly, IL-36 might actually be able to look at commensal to pathogenic shift. So quite often our commensals are sitting there quite happily, and they may take on a virulence factor or uptake a plasmid, etc., and then become pathogenic. So it's only when they become pathogenic that the IL-36 would be able to sense this by the actual necrosis of the cells and then the actual degradation by various different proteases. So obviously it's very important in pathogenic disease, but I suppose in terms of inflammatory disease, then having this good molecular understanding of how IL-36 works might be quite important for actually inhibiting in various different inflammatory diseases. So also in various different inflammatory diseases, maybe in eczema, but certainly in certain types of psoriasis, they're very strongly correlated with infection. So people used to think, oh, well, these are just autoinflammatory diseases which just are initiated randomly. But this would be a great mechanism in which a particular microbe or a microbial entity is the initiating trigger or factor for some of these inflammatory diseases. So guttate psoriasis, for example, is very strongly correlated with strep infection. So a number of weeks after having tonsillitis, certain individuals that are susceptible to psoriasis will get a big eruption in guttate pustules all over their body. And this is very strongly correlated with strep infection. And now we've shown a potential causal link. This could be the SPEB-B protein, which is activating IL-36 and causing this autoinflammatory response. So this is a good link between microbial triggering and also autoinflammatory diseases such as psoriasis. And it's also been shown that IL-36 is upregulated in inflammatory bowel disease, and it's also associated with an inflammatory bowel disease is often associated with microbial dysbiosis. So when you get the wrong types of bacteria in the wrong place, and when they start invading the epithelial cells of your large intestine, etc. Okay, so that just finally leads me to acknowledge all of the people that have sort of done this work. So as I say, most of this work was done by Tom in my lab in close collaboration with Miriam Whitman. And we've also had some collaborations from groups in Manchester in the UK, from Germany, and also from Japan as well. And finally, thanks for listening.
So are there any drugs in the market which are already inhibitors of IL-36 for inflammatory diseases, or is this a new therapeutic target?
It's a relatively new therapeutic target. So there are examples of the IL-1 receptor antagonist, which are involved, as it's called anakinra, which blocks IL-1 activation. And people are certainly looking into using the IL-36 receptor antagonist. So as I mentioned earlier, there are defects in certain individuals where you don't have the IL-36 receptor antagonist. So this is a negative regulator. And these individuals will suffer from generalized pustular psoriasis. So people are looking into using IL-1 receptor antagonist to actually treat these individuals. But there are some patented antibodies, not against IL-36 itself, but against the IL-36 receptor, that are now in various clinical trials for inflammatory diseases. And these may well be slightly more specific than IL-1 antagonists, in that if you're just looking at the very surface of epithelial barriers, then you might be able to knock out IL-36 function and prevent certain triggering of certain diseases. Whereas if you knock out IL-1, which is found in lots of cells throughout the body, then you might expect to have more side effects from that. So yeah, certainly people are investigating it, but there are no actual FDA-approved drugs as yet.
It's very interesting. It interested me mainly because it is such an early response, and also because it is so available, because almost all the pathogens do have proteases inside. Very interesting work.
Yeah, I think that's a clever system. That in fact it has this flexible region which is highly susceptible to proteolytic degradation.
Yeah.
But normally it sits inside the cell, and it's absolutely fine and non-inflammatory when it sits inside the cell.
Yes, I also feel it's quite clever because it needs to be activated by proteases, and you can easily imagine that, for example, our epithelial barriers can be damaged by other factors, not exactly pathogens. For example, I don't know, we eat something hot or spicy, and our epithelial tissue is damaged, and then it would make no sense to make this whole inflammation just because we ate something hot. And it's good to know how to make this distinction between something pathogenic and something just random.
Yeah, definitely. I mean, I should say that in certain circumstances where you get a massive upregulation of IL-36, then you actually can get activation in the absence of bacterial proteases. So for example, in psoriasis, IL-36 is one of the most upregulated genes. And if you cause trauma by itching or rubbing, then you can actually release proteases from host cells. And these aren't as active as the proteases from pathogens, but they will indeed, when you have a large number or a large amount of IL-36, they will actually activate IL-36 and cause inflammation. So people with psoriasis, they often get it in the same place if they're wearing a handbag or a strap on one particular shoulder. there's a Kerbner phenomenon in which this sort of micro trauma is believed to actually cause some sort of activation. And our take on that would be that the microtrauma causes, the bursting or rupturing of cells, the actual release of some of the proteases or lysosomal proteases from the cell, and these can in fact activate IL-36. So when you have high levels of IL-36 when you shouldn't have, and then you have microtrauma, then this can also activate inflammation as well.
Yeah, I also like that there are so many steps until the inflammation really begins. We have a question from audience, so from YouTube. And
okay.
There's a question about psoriasis. Why is there a delay of weeks after strep infection for psoriasis to show up?
Well, no one has truly investigated that. At the moment, this is just a correlation, but it could be that the proteases are released within the throat. They need to get to the skin where they can actually have an effect. So yeah, it is surprising that it does take that time, but maybe this is after onset of the actual disease. Maybe you need a critical threshold of the actual protease itself to actually get to the epithelial cells. And again, this is only in certain individuals or people who are susceptible to guttate psoriasis. So most individuals who get strep throat will not get guttate psoriasis. So I appreciate it's rather hand wavy, but I think it must be a threshold thing in terms of you need a certain amount of the SPEB protein released, and it needs to get to the skin as well.
I wonder if it's also because people who are getting autoinflammation, like in case of psoriasis, clearly some part of their regulation doesn't work as well. So it may take some time for this inflammation amplification to take place to a point where it's out of control, and now you have a very bad rash on your skin.
Yeah, certainly. I mean, this doesn't happen with all people. So you must have some sort of genetic susceptibility for this to be triggered. So yeah, maybe they do have inherent problems with, I don't know, their barrier defect or something which actually allow over time for this protease to get there.
Yeah.
I mean,
oh yeah, Martin, continue.
Yeah. So as I say, at the moment, this is just a hypothesis, but it's a nice correlation anyway.
In psoriasis, there is one hypothesis that some antimicrobial peptides, and you just showed that IL-36 does induce antimicrobial peptide secretion. So one of this antimicrobial peptide called IL-37, I think it's a cathelicidin class of antimicrobial peptide, and it's positively charged. And what it does, it binds to the negatively charged DNA of bacteria, and it forms this stable complex, and then it goes and triggers TLR 7 and 8, or even 9, and that further increase. That's an amplification step at this point. So I wonder that might be also something.
I mean, yeah, we have actually shown that LL37 and DNA complexes are taken up by keratinocytes, which are obviously in the epithelial layer. And I think, yeah, LL37 is certainly increased by IL-36. And again, obviously, this is obviously quite important in lupus, etc. But I'm not sure whether IL-36 has been shown to be massively increased in lupus. It's most dramatic in. It could well be, yeah. Yeah, so it's been most upregulated in psoriasis. It is shown to be upregulated to a lesser extent in eczema, and there are some reports of it being involved in lupus as well.
Okay, we have another question from audience in Facebook. Chrissy says, thanks for the talk and discussion. Do you think there is a premise for using protease inhibitors as treatment for autoinflammatory diseases during microbe-associated flares?
I mean, we've certainly considered that. I think one of the problems is that, as we discussed, you have such a broad range of proteases that can actually activate the IL-36. So you'd probably require a whole cocktail to inhibit a number of proteases. So I think that's why the most logical route would be to target the IL-36 itself, or as people are doing, targeting the IL-36 receptor
Mm.
via monoclonal antibodies. But yeah, certainly groups have considered that. My personal opinion would be that it would have to be quite a broad specificity, and you'd have to inhibit, a lot of the proteases for this to be, functional.
Mm.
Do we have any more questions from the audience?
Not right now.
Okay, so I can put my one point that I was waiting for. So this looks like a 3 checkpoint system here. That first you need something to increase the expression. Next you need something to get the IL-36 gamma outside, and then you need something to cleave it. So it's a very well regulated system, and in normal circumstances it should not be creating havoc in our body, right?
Yeah, that's our general hypothesis. I mean, as inflammation can be so damaging, and you can have a lot of collateral damage, then obviously it's very important to regulate it. And on top of that, you also have the IL-36 receptor antagonist, which is often upregulated during inflammation as well to help as a negative feedback loop. So yeah, so you have yet another level of inhibition or regulation there as well.
Did you say the receptor antagonists are made by the same cells?
They are, yes.
Okay.
And often during inflammatory insult, you'll actually see an increase of the actual receptor antagonist as well, as a negative feedback loop.
Okay. What is the next step for you guys in this project?
Well, unfortunately, we were hoping to do some animal studies, but due to the funding situation, we're now actually moving on to pastures new.
Wait, they're not funding this study? If I had money, I would pour in $1 million today.
Well, if you've got $1 million, then feel free to email me.
That's probably why I don't have money.
You're too generous.
Yeah, no, but this is a very nice study. I liked it so much. I always say in journal clubs, because there are certain papers that you read, you take some home message, and you never forget it, because that becomes a crucial part of your concepts. It revolves around immunology generally. So this is one of the concepts that I'm going to keep with me for a very long time.
Oh, that's good. So it should be said that this isn't the only system that actually senses damage. Right. So there is the IL-1 system as well, IL-1 alpha, which is a bona fide alarmin as well. So,
but IL-1 alpha is not secreted as a proform.
No, it isn't. But it's in the cell, and it's only released upon rupture.
Okay.
So our group shouldn't take full credit for the concept of alarmins or danger signals. Just right. A little bit.
Well, I like this protease sensing part. That's very cool. That you know. And actually, there's a philosophical question. You're saying that there are some commensals that don't make proteases, and there are these pathogenic organisms that do make proteases. What are the commensals losing when they don't make proteases? And what are they gaining?
Well, I mean, I think the ability to invade cells. So I suppose for proliferation of cells or of the actual pathogen, you need nutrients, etc. So you need to invade the cells. You need to damage the extracellular matrix, etc., to allow more tissue fluid, etc., to come into that environment. So I think for pathogens to invade and to proliferate, they need extra nutrients, and that's possibly where their requirement for proteases come in. Whereas commensals are probably sitting there in a nice, happy equilibrium on the cell surface or on the mucus or on your skin, etc. And again, if they become pathogenic or problematic, and they start proliferating too much, then they will then start causing damage, perhaps.
There's this. Have you guys heard of Prisoner's Dilemma?
No.
So it's a game theory that when in a system whenever there is cooperation, everybody gains. But if there is one party that is not cooperating, they gain more than the other party that is cooperating. So there's always an incentive to deceive the other party. So I feel like in this case, the pathogens always have this incentive that they can get more out of their host by deceiving, or they could play well with the host and both of them enjoy the same rights. It's just some weird things I think about in my free time.
Okay.
All right. So do you guys have
But
any other questions? Oh yeah, Tanu.
Jatin, just to add on to your previous point, the commensals generally are in their stationary phase and they only grow or come back to their, go to the log phase when, at least for the gut bacteria, when the person is having food, they get the nutrients from there and then they replicate or they actually like survive, but otherwise they're in their stationary phase and they're mainly in the lumen, so they do not go to inside the core of the gut or into any cells and infect. And that is one of the mechanisms through which they also are in a equilibrium and not in a very, very high population. But I think the main motive of pathogenic bacteria when they infect a person is to replicate continuously so that there are more of that bacteria or any pathogen, because when they come into a body, they're in so low quantity, their main motive is to keep replicating. And that's where a major difference comes in.
Right. And in the gut, a lot of our IgAs are tagging these and keeping them from crossing the barriers. So
Yeah,
the host itself is also making effort to keep them out
they are
where they
mainly
shouldn't
in
be.
the mucosal. Yeah.
Yeah. Okay. If you guys don't have any other question, I think this would be a good point to end the discussion. Thanks a lot, Martin, for joining us today. This was a
Thanks for
very
listening.
nice discussion. I loved it. New concept for me. And everybody who is watching on Facebook or YouTube, thanks a lot for joining. The video will stay on the live stream, so you guys can go back and watch it if you want, or can share it with others who would like to, who are interested in knowing how our bodies differentiate between commensals and pathogens. For now, I'll see you all 2 weeks later. Bye bye.